Okay, so here's, more on the real power of these metabotropic receptors or these G protein coupled receptors or GPCRs. They can have significant impact over the actual physical structure and sort of maintenance of that particular synapse and that particular cell. And this is really how a lot of really important communication between what's happening in the synapse, in the cleft, like how much stimulation is occurring out here. And we're going to see drugs have a significant impact sometimes on the amount of stimulation of receptors, both presynaptic and postsynaptic. And that those actions, those drug actions here can have many downstream effects. They can ultimately strengthen particular synaptic connections or maybe weaken other synaptic connections. And how do you do that? How can a drug, what's happening out here in the synapse, influence the physical structure of the synapse, right? And of that cell? And really, it does that because what's happening here can be communicated back to the nucleus of the cell, to the nucleus in the soma, to the chromosomes, to the genetic material. And it can result in changes in the expression of genes. So the transcription and translation, of proteins, right? So it can change what's happening in the membrane, like what receptors are actually found here. And that's one of the real huge influences we're going to see about, drugs on, on synapses, is that they can actually alter the physical structure and thus functioning of those synapses by communicating back to the nucleus and changing gene expression. Well, how do they do that? Well, there's stimulation of these metabotropic receptors. So here's a G protein on the inside. My head's the nucleus, right? So, and then out here in the cleft, the neurotransmitter binds, or a drug binds. Let's say it works the same way as the neurotransmitter. We call that an agonist drug. We'll tell you this later too. But basically, it binds, changes shape, right? Releases the G protein. And we said before, right, the G protein could go over, bind to a G protein gated channel, open it, depolarize, hyperpolarize, do that, right? That's fine. It can do that. But it could also come over here and interact with another protein. And this other protein, there's one indicator here that's sort of embedded in the membrane too, but it'll change its shape, right? And it will release something of its own. This is called this second thing that's released. So what's interesting is the binding of the neurotransmitter extracellularly, to the metabotropic receptor, releases the G protein. The G protein is sometimes referred to as the first intracellular chemical messenger, right? It's carrying a message that there was activity out here in the cleft, right? A neurotransmitter or a drug hit me here, right? And now I'm the first intracellular messenger. It can then bind to something else that can change its shape and release something else. And that second thing it's releasing, there's a very commonly released chemical called cyclic AMP, but this is a second intracellular chemical messenger. And then this second messenger can then go interact with another protein, that's found inside, this postsynaptic cell, right? And it can change its shape, and it can sort of activate it. It has a new functional role now that it's, been impacted by the second messenger. We call this the third messenger. The third messenger can go off and bind to another chemical, right? And that can influence its structure and thus function. And that's the fourth messenger. And so you have this kind of stream of, intracellular chemical changes that are set off by the binding of the neurotransmitter out here in the synaptic cleft. They call this a biochemical cascade, that results. And, downstream, the third, fourth, fifth, sixth messenger, some of those activated chemicals can enter the nucleus of the cell. I'm going to my head, right? And basically bind to the chromosomes and, unzip certain segments of your DNA, for transcription and thus translation and the building of new proteins. So it can what we say upregulate the expression and the building of specific receptors, for example, or transporters or enzymes or channels. So it can increase, upregulate, the production of certain proteins, or it can actually bind to certain segments of the chromosome, right? Some of these downstream biochemical messengers, right, that were, set off by the binding of the neurotransmitter out here in the cleft. And it can zip up sections of the DNA and reduce the expression of specific genes, what we say down-regulate the expression of receptors or channels or transporters or enzymes, other protein machines. So be aware that at all your synapses, there is some level of synaptic, chemical communication. And this is important because there are always biochemical cascades, communicating back to the genes to, inform, the nucleus, about what's happening out here in the cleft. What do I need? What other proteins do I need? Do I need to upregulate expression of this? Do I need to downregulate expression of that? And there have to be constant, replacement and adjustments, to maintain the integrity of all the synapses that we are going to maintain, basically in our nervous system. Because these protein machines are literally machines. They're moving, they're changing, they break, and they have to be removed and replaced, right? So there's got to be communication back and forth along these biochemical cascades. But drugs are going to have a big impact because they can, sort of dramatically change maybe the level of activation, at these metabotropic receptors, for example, which can dramatically increase, the activity along the biochemical cascade, which could have, bigger influences, let's say, on the expression of genes, right? Or we're going to see some drugs are going to actually not act as what we call an agonist and act like the neurotransmitter and, overstimulate, but some of them are going to block, some of these metabotropic receptors, right? They're going to act as what we call an antagonist. They're going to antagonize the metabotropic receptors. So you're not going to get the release of the G protein. And then you're going to have, a real drop in, biochemical communication, like that cascade communication back to the genes. And that will also provoke changes in the zipping and unzipping of different segments of the chromosomes, and changes in gene expression. So I think this is rather powerful, experiences, drugs, the level of activity along these different synapses has great influence over their physical structure, which genes get expressed and to what degree. And we're going to see actually some of these changes, it's rather fascinating, are rather long-lived. Some of the like overactivation or underactivation, particular biochemical pathways can attach, chemicals to the chromosomes, right? That can sort of more permanently switch off, the expression of particular genes, or more permanently switch on the expression of particular genes, or more permanently alter, the level of expression, and that can have, significant impacts on the physical structure